I think we'll get started. I think we have quite a full house plus, so thank you all for coming. So Dr. Raymond and I are really thrilled to chair this workshop. So what we're planning to hope today is to share with you this list of remarkable speakers to highlight their efforts and progress in their clinical trial research. So the goal of this workshop is to present the latest clinical trial research, especially the early phase trials and other interventional and prospective trials, to which some of you may have screened, enrolled, and referred patients for participation as well. So today we'll be learning about, again, early phase trials, including genetic research, a use of ETI in alternate populations in terms of effectiveness and safety, and also innovative imaging for as a useful outcomes measure. So we'll remind you to please, we have plenty of time for questions after each speaker, so please come up, if you will, for questions and discussion. There's also, as you know, the online forum to submit your questions. We'll be screening and reporting as well. And please silence your cell phones. Anything else? All right.
I think that's it. This is going to be a great session. This is a full room.
So we'll start, please, with Dr. Gilly Hart. She is CEO of SpliceSense in Israel, and this is a company who specializes in mRNA research. And we very much appreciate your coming today. Understand the lengths it may have taken to come to travel at this time. We're very much looking forward to hearing your talk.
Thank you very much. Yeah, I think that's fine. Presentation.
Yeah. The start button.
Yeah.
Right there.
Oh. So I would like first to thank the committee for the opportunity to introduce our work. And again, I would like to dedicate this talk to my brothers and sisters, 1,500 babies, toddlers, men and women, young and old, that were raped, tortured, and murdered, and also to more than 250 young and old people, kids and babies, that were kidnapped and are being kept hostages in the Gaza for more than 4 weeks now by the Hamas-ISIS terrorists. So today, I'll be discussing SPL84, antisense oligonucleotide for treatment of patients who carry the 3849 mutation. And this talk will be focused on the safety and the toxicological work that we have conducted in order to move forward and run our phase 1 and phase 2 study. The phase 1 study was completed successfully, and you are more than welcome to look at our poster. And just before I do that, and again, I would like to briefly introduce SpliceSense. We are a pulmonary-focused company utilizing antisense oligo platform technology for treatment of those pulmonary diseases. As you can see in this slide, we are focused on specific unmet cystic fibrosis mutations, but also on large and still unmet indications. Just to remind everyone what antisense oligos are all about, we are talking about very short RNA sequences that specifically bind using a Watson-Crick interaction to the target region on the mRNA or pre-mRNA in the nucleus or in the cytosol, and by that modulate the target RNA and also affect the protein properties. It can either decrease production, it can restore production, and can overcome mutations. We are focused on the 3849 mutation. We're talking about around 1,500 patients globally. For those patients, there is no specific approved treatment. However, in the US, the Kalydeco and the Symdeko are approved, and we do know that they do have very modest to poor improvement in the lung function. So when considering the benefit versus the cost, it was concluded in Europe that those drugs are not approved for those specific patients. So what is this mutation? This mutation really leads to an insertion of an abnormal sequence, a crypt exon, to the CFTR mRNA, leading to a non-functional CFTR. Our antisense oligo inhibits the insertion of this specific crypt exon and generate a fully functional CFTR. And this is very important to emphasize because we are focused on generating a wild-type CFTR. And as you all know, you don't need a lot of fully functional wild-type CFTR to really generate a significant improvement in the clinical outcome or in the lung function. We did a lot of work trying to really evaluate the activity and the pharmacological effect of our SPL84. For example, here you can see some studies that were conducted by Professor Isabelle Saramite from the NACAR Institute, in which her lab evaluated our ASOs using H&Es or HbAs from different patients, homozygote, heterozygote. And in all of those studies and other studies, it was concluded that the treatment with this specific antisense oligo, SPL84, leads to full restoration of the CFTR activity using the industry standard, the Using assay. But again, today I would like to focus on the safety and toxicological outcome of treating with this specific antisense oligo. And when we think conceptually about what is needed in order to really evaluate the safety before we move into humans, or more specifically and more importantly, before we move to treating patients, there is a set of regulatory guidelines which is needed to be actually established. And when thinking about antisense oligos, I think that the guidelines which were generated in general by FDA or EMA are slightly less mature because those are not as common as other compounds or drugs. It's kind of a cutting edge. So we really needed to work closely with both FDA and EMA in order to structure this safety program and to make sure we are covering every aspect of it. And I want to share some of this data. I would like to start with the safety assessment. And when thinking about safety assessment, there are two aspects and two questions that need to be addressed. The first one is, since we are delivering an antisense oligo that binds to the CFTR, do we modulate any other mutations? Do we have effect on wild-type CFTR? Because if we do, obviously it can potentially generate some safety concerns. So we did a lot of studies trying to address that. And here, for example, you can see a study that was done in healthy volunteers, non-CF patients, H&Es. And we were able to confirm, as you can see, that when we treat those cells at high doses with our antisense oligo, there is no modulation or no effect on a fully wild-type CFTR. And therefore, there was no effect on the Using or the CFTR activity in those H&Es that were collected from healthy volunteers. And this is very important. On the other hand, we also tested the effect of our ASO on other mutations. And we have done that to a variety of mutations. You can see here H&Es that were collected, for example, from F508del patient. And we were able to confirm that indeed there is no effect, no improvement, obviously, but no interference, not with the RNA and no with the Using activity. So this is really the specificity effect of our ASO. But another aspect that needs to be addressed is the off-target effect. And this is also very important, and this is one of the major concerns of the regulators. Because when thinking about off-target effect, we are talking about very short sequences which can potentially have an homology to other non-related genes. And we had to evaluate whether our ASO, which is 19 bases long, has any interaction with non-specific genes or RNA in the periphery and, most importantly, within the lungs. The concern is that it might bind to different regions and can modulate those regions, whether it's splicing, whether it's interfering, and we had to do that. And it was done in a two-step manner. The first part was to do an in silico analysis. It was conducted by a vendor that is quite experienced with those studies and can do it to support regulatory submission. And in those analyses, we got a list of potential genes in which are considered to be primary or mature RNA, meaning introns, exons, mitochondrial, DNA, RNA. And in those analyses, we tried to identify potential sequences in which our ASO can bind to. The outcome of this analysis was that there were 7 genes in which we had homology with 2 or 3 mismatches. There were no sequences, whether those introns or exons, that we had a full-length homology or even 1 mismatch. And this is really critical because it really affects the binding. But it's also about the location. Are those mismatches at the middle? Therefore, they will interfere with the interaction. Are those at the end? And we did this analysis, and we did a very robust risk assessment that concluded that considering the fact that all those sequences were within the intron, the deep intron of those genes, and all those genes were genes within the periphery, there is no concern, which was confirmed by the regulators. But there was one gene that we really wanted to further test, and this is the NET4 gene, as you can see here. It's one commonly that you can find in the lungs. And we did an in vitro analysis where we took H&Es, HBEs, we treated them with our ASO, and we evaluated the effect of our ASO on those specific RNAs. And as you can see in this figure, there was no impact, no modulation, which concluded that there is no off-target effect. Another study that was conducted was really to evaluate the interaction of our ASO with commonly used drugs in a CF setting. You can see a list of 3 commonly used drugs, for example, pulmozyme, some antibiotics. Our ASO was spiked with those specific drugs, and we evaluated the interaction, whether there is interaction, whether our ASO is now being degraded or anything like that. And the conclusion was there is no effect on our ASO or on those drugs when they're being combined with those specific drugs. Although our ASO is targeted for inhalation only once a week, and though some of those drugs are given more frequently, this is something that needs to be addressed because it might have a longer half-life in the lungs, it might get interacted, and we have done that. And lastly, before moving into animals and running toxicological studies, we really had to confirm that the integrity and the activity of our antisense oligo is maintained through nobilization. Our drug is given by nobilization to patients. It was given to those animals by nobilization to mimic the route of administration. And through this nobilization, through the process, there are some shear forces that might potentially affect the ASO. We had to make sure that everything is maintained intact, and we have done those studies, and we evaluated the purity. We made sure that the impurities are not affected, and we made sure that the identity, the footprint of our ASO is maintained. And lastly, we have developed and qualified a potency assay that confirmed that following nobilization process, our ASO is still active as it was before. And this was done before and after nobilization for all our studies, starting from the tox, moving to the human studies. So those studies were really related to the safety, but now I want to take you and talk about the actual studies that were conducted in animals. And when thinking about those studies, first we had to confirm that our ASO can get to the lungs and can be properly distributed, just to make sure that when we say potentially, and we do, that our ASO is safe, we know that it gets to the different regions, the different cells in the lungs. And this study actually was done to confirm that, to confirm proper distribution. We treated animals, in this specific case, both beta-ENaC muco-obstructive and wild-type animals with our ASO. We then, by hybridization, stained the lungs of those animals, and were able to confirm proper distribution of our ASO within the lungs and within the different regions and cells within the beta-ENaC, which were comparable to the wild-type, confirming that indeed our ASO is properly distributed in mice. And on top of that, we were able to confirm the long-acting properties. Because when we did a study in wild-type animal and treated them only once and sacrificed those animals 4 weeks later, we still had a very robust response. The same study was done in monkey in preparation for the toxicological studies. We were able to confirm proper distribution of our ASO within those lungs, within the different regions of the lungs, and most importantly, within the target cells that we are aiming to modulate, for example, ciliated cells and secretory cells. So once we felt that we are ready to move forward and we have all the data to support our studies, we started a battery of toxicological studies. The first part of those studies were named safety pharmacology studies, in which we evaluated, following treatment by inhalation with our ASO, the different origins or systems within those animal bodies. We evaluated the neurological effect, potential neurological effect of our ASO, and we evaluated the effect on the respiratory system, This was done in monkeys that were treated at different doses, up to 20 mg per kg per week with our ASO by inhalation. And as you can see, there was no effect. We ran a very robust and precise cardiovascular study, trying to see if there are any effects. Maybe our ASO gets there, can interact with something. And then we did some in vitro studies in order to evaluate the potential genotoxicity of our ASO, because it might interact with some chromosomes and some other parts of the DNA. And this was also concluded to not to be an issue. The next step was really to move and to run the toxicological studies. And here I want to kind of pause and talk more conceptually about inhalation and safety concerns. So when thinking about the safety profile or the toxicological profile of inhaled drugs, or more specifically inhaled antisense oligos, there are two main issues or two main findings that were reported in the literature. The first one is the actually accumulation of alveolar macrophages. When you see that in the pictures or those histopathological photos, you see those big macrophages that really uptake the oligos or uptake the drugs. They look very fat, very big. And when considering the fact and the historical safety data, this is not considered as an adverse event. This is really the classical clearance mechanism of antisense oligos and any other RNA or compounds from the lungs. The macrophages uptake it, process it, and then it's cleaned through the lymph nodes to the periphery, and then from there it goes to the kidney or to the liver. But the second issue that you might see when thinking about treatment of lungs with ASOs is really infiltration of mononuclear cells. And this is considered as a severe or I would say even an adverse event, because it might imply that those mononuclear cells are actually cells that are triggering inflammation that can lead to a severe side effects in those individuals' lungs. So basically, when you're running toxicological studies, those are the two main things that the histopathologist team will look for. When they see those alveolar macrophages, they say, well, I don't see any issue with it. But if they start seeing infiltration of mononuclear cells, this is a red flag. And this is what we are looking in our studies. When thinking about previous programs of antisense oligos, I would like to actually focus you on this table. Those studies, those programs moved into the clinic. Some have completed phase 2 studies. Unfortunately, none of them matured or advanced, but this is not due to safety concerns. It all goes back to efficacy. In all those clinical studies that antisense oligos, different kind, different sequences, different mechanism of actions were given to those individuals, to those patients, the safety profile, which was evaluated and monitored, was good and promising. So this was not the issue. But we do need to remember that the lungs are really kind of a black box, and we need to really pay special attention because of that, and those toxicological studies are really critical. They are predictable. Another thing we need to remember, and this is well established, is that when running those toxicological studies, whether you're doing that in rodents or in monkeys, the rodents are much more prone to develop inflammation. They're considered to be much more sensitive. And therefore, if you do see something, it is suggested it might not be the relevant model or it might not be an issue, but you need to continue and follow it and make sure it's reversible. So this is kind of a very brief introduction, and I want to share our toxicological data with you. The first study that we conducted was, again, GLP, Good Laboratory Practice Study. It was a 9-week study in rats. And in this study, we dosed those animals up to 54.4 milligram per kg. And all of the microscopic changes that were evaluated, whether it's lung, lymph nodes, and so on, were considered non-adverse. And the NOEL was really the highest dose that was given and actually quantitated in those animals. Here you can see some figures that actually illustrate that. We see there is alveolar macrophages, which can be seen. They're big, they're very swollen, but other than that, there was nothing, and it's not considered adverse. In the 9 weeks tox study in monkeys, the highest administered dose was 20 milligram per kg. And again, on top of that, we evaluate other elements related to safety. But again, there were no microscopic changes in the lung or in other parts of the lung, including lymph nodes, and therefore it was also considered non-adverse, and the upper safety dose was the highest administered dose. We also evaluate the systemic exposure, and as expected, the systemic exposure both in mice and monkeys was very low. Most of the drug, ASO, was maintained within the lungs in all doses. And when we start to look, okay, so what are the other target organs in which we can really quantify our ASO, we're able to see that, again, indeed, the majority of the drug was within the lungs, but we're also able to detect some of it in the kidney and in the liver, because as expected, this is the classical clearance of those ASOs mainly through the kidney. But there were no safety issues or no functional effects on those organs. So based on this data, we moved forward and successfully completed the phase 1 study. And now we are headed to initiate our phase 2 study early next year in CF patients. And with that, I would like to thank the team, our collaborators, and all the consultants that work with us on this program. Thank you very much.
We'll open the floor to questions.
Thank you for a nice presentation, and thanks for coming to us to talk to us in these difficult times. Did you test whether the ASO are amenable to breakdown by neutrophil elastase and other components of CF sputum which might be relevant for dosing it by inhalation?
Yes, we did stability studies in hyperconcentrated CF sputum, and it was, as of today, it was stable within it and kept its integrity up to 72 hours.
Thank you for a great talk, and as Felix said, thank you for coming in these troubled times. I was a little surprised to hear you describe the lung as a black box, because it's a black box with a great big opening, namely the airway, and there's plenty of literature about using in humans, about using things like induced sputum, like breath profiles, like exhaled breath condensate and spectroscopy, which would seem to me you could be using if you were trying to detect an inflammatory response developing in the human studies. So maybe it's not quite as black as...
I agree with you, and all those are incorporated within our study design, but still when thinking about injecting a drug peripherally versus lungs, I think it's much easier to evaluate safety concerns and issues early on as opposed to lungs, which might be developed later on. I think that's a major notion, and by the way, this is really the concept when thinking about FDA feedback or EMA feedback when they think about lungs as and the potential safety issues.
So there is that window that you can really get a handle non-invasively in children as well to use. I'd have a look.
I have one question. You talked about testing with common inhaled medications in CF, so Dornase or antibiotics. There was some data presented yesterday out of Iowa showing that hypertonic saline potentiates adenoviral transfection, which was fascinating. Did you look at hypertonic saline and whether there could have been an impact on stability or I mean, even efficacy?
So we did kind of what we call stability study under different conditions, including salts, pH. We didn't specifically test the hypertonic saline, but we did test saline under different extreme pH and condition, which might cover that. But that's something we can consider as well.
Any other questions? Thank you very much.
Thank you.
Okay, our next speaker is Marty Solomon from the University of Alabama at Birmingham, who is going to talk to us about ETI and N1303K mutations. So, Marty, very excited about this.
Thank you. As this is Keely, I want to say thank you to the organizers for the opportunity to present this, and thank you all for coming into this little cozy cottage here we have for this workshop talk. So appreciate you all making. Let's see, I think I've clicked the wrong.
That is not your talk.
Yeah, it's odd that it didn't come up right. Yeah. I think I've clicked it. Let's see. All right. Thought I did that the last time. Great. All right. Wonderful. So now I'm able to get started again. Thanks for the opportunity to present our work. I'm presenting on behalf of a large collaborative group, but I'm have the fortunate opportunity to do this. I want to say I saw a quote, I think, by Emerson recently that ended with the concept of you've lived a good life if you can help at least one person breathe a bit easier. And this study has been one of those that's sort of given me that feeling. So I hope you all will be left with that same concept as I present the work. So this title was interim results. However, I'm pleased to say that we've finalized this trial with the completed enrollment. So this will actually be the final top line results of this study. So let's see if I can learn this system again in here. So these are my disclosures. I want to start with the acknowledgments. This work took a village. Mostly the patient participants who, as you will see at the end of the talk, flew from literally all over the country. We had many that were willing to fly from all over the world to promote this work. I really want to thank my co-PIs, Rachel Linneman and Eric Sorcer. They righted my ship more often than not. And the research coordinator team has been incredible for this, especially our leads at both sites, Emory and UAB, Rachel, Heather at UAB, and Ashley at Emory, and others that have supported this work, and then my lab and other collaborators, and then really the funding sources. This was a pet project by the CF Foundation added on to an R01 project, and so I really appreciate the funding opportunities for that. So wanted to leave at those. So the premise of the collaborative work that's been ongoing for several years, both at Emory and UAB, has been really improving understandings of mutations and CFTR variants, developing an in vitro system which was focused on in a workshop by one of our collaborators in Brian Davis's lab, the iPSC ALI-derived model, which I will not discuss today, and then pairing this with feasible clinical trial methods that can understand patient responsiveness to drugs in rare mutations, and really hopefully comprehensively evaluating these mutations to hopefully gain improved access or at least better understanding of these mutations for patients. So I'm going to turn to the N1303K story. And so in vitro data has previously indicated that this may be a responsive mutation, but however, as many will be aware, in the Vertex FRT system, this mutation or variant had almost no activation in that system. So therefore, addition of this mutation on off-label was not achieved by the FRT system previously. However, the N1303K mutation is the second most common class 2 mutation behind F508del, and it's a folding defect very similar in the NBD2, which has a similar gating mutation type defect on the cell surface. And there's been a significant amount of theratyping work by various labs and various cell systems, including some data that I'll show in a moment from our work, that N1303K will be activated by various potentiator and corrector combinations. This is data from Eric Sorcer's group, who's been developing a companion FRT system to the Vertex system, which is slightly different, which we can see when we have the far right-hand box, we have triple combination or ETI, that there is some beginning to have appearance of kind of a blurred, more mature form of this protein, not what we consider a significant activation like with F508del, but there is the appearance of that increased activation and expression of a more mature form of N1303K when you have ETI on board. This is data in collaboration with both John Brewington from the Theratyping National Resources Center and then our labs at both Emory and UAB, showing that in both this same FRT system and in nasal cells derived from a patient with N1303K and a second minimal function mutation, that there is in fact fairly meaningful increase in CFTR activation with triple combination in these cells exposed to this in an Using chamber environment in both of these systems. Finally, we derived iPSCs from a patient who was being enrolled in our companion study looking at rare mutations that Rachel and I have been co-leading as well. And we had a patient was enrolled as kind of a negative control with an N1303K minimal function CFTR mutation. And these patients had their iPSC-derived ALI. And similarly, we found activation of the CFTR function in those cells. So this kind of complemented in our hands in various cell systems that there was possibly meaningful activation in many of these patient usable in vitro biomarkers for CFTR activation with CFTR modulators. So this allowed us to utilize a trial design, which I had spoken about a few moments ago, and Rachel Linneman outlined yesterday in a rapid fire poster talk discussion using in patients with rare mutations that had partial function phenotype with them. And so we sort of borrowed this clinical trial design in which we had a prospective 2-center open label study or trial and 20 subjects who were eligible with CF encoding an N1303K and either another N1303 mutation or a minimal function by the Vertex list second mutation in 12-year-olds. And basically the idea of this trial design was that patients would be exposed to ETI by standard dosing for a 28-day period with observation, and then they would have a washout period in which they would act as their own control to see if they regress back to baseline findings in these clinical and CFTR activation endpoints. And the hypothesis in this study was that ETI will restore sufficient CFTR function in the N1303K mutation to improve sweat chloride, and then that would lead to improvements in key clinical endpoints like lung function, et cetera. We'll also test whether these changes correlate with observed return to baseline at the washout period, and then whether these track with the iPSC data, which I will not be talking about today, will be a future result of this study, we hope. The endpoints of this were, the primary endpoint was absolute change in sweat chloride from baseline through day 28 and correlative analysis of the iPSC data, which is pending at this time. I will not show that data today. The secondary endpoints for efficacy with lung function, CFQR, and weight changes through day 28. And of course, safety was monitored as would be with any routine clinical study. So this study is fully enrolled, and all subjects have completed through day 56. 22 subjects were screened. 2 of those were ineligible due to exacerbation at the time of screening, but 1 was able to rescreen into the study at our site. This is the baseline and demographics of the patients. You can see these are pretty similar to a routine modulator study for a classic cystic fibrosis population, mean age of about 25. About 60% were over 18, but there were some in the 12 to 18 bracket. At the time we started this study, there was not the approval for below 12-year-olds, so we've only enrolled up above 12 at the time for this study. We'd hope to do a study in younger kids in the future, though. There wasn't a good distribution of male and female patients. We did hope to enroll a more diverse patient population, given this mutation is a bit more prevalent in certain racial and ethnic groups. However, we only enrolled 2 patients of other race, but there were 2 subjects of Hispanic or Latinx background. So a little bit more diversity has been seen in some other clinical trials for CF. All of these patients, unlike what we had hoped, were N1303K minimal function mutations. We did not have any homozygotes, but as you'll see, there's interesting data from other labs about the homozygote group. These patients had a pretty typical CF complications history and a fairly usual distribution of baseline predicted lung function and a sweat chloride mean of about 109. So a very classic cystic fibrosis patient. For interest, the minimal function mutations which were enrolled or listed by their distribution at the bottom. So many of these were PTC type mutations or other very minimal function type mutations as the second allele. So as you can see, unfortunately, few subjects had substantial reduction in sweat chloride at day 28, sort of refuting our hypothesis in this data. But and as you can see from the responder analysis, very few had had an increase of sweat or decrease in sweat chloride that would sort of be over the thresholds associated with Symdeko or earlier dual combination modulators. However, I'm going to say it's a big but. The absolute change in lung function was remarkable, plus 9.5% for this population. And this mirrored significant improvements in CFQR with a greater than 20 increase in CFQR, and then fairly meaningful weight changes in this population at a short endpoint. As you can see, the secondary clinical endpoints, almost every patient was a responder across this group. And so we were left with thinking this was a remarkable clinical response. In addition, as I will remind you all, these secondary clinical endpoints regress back basically to the patient's pre-baseline means in the group. So that taught us that we thought there was a significant drug effect rather than this being some sort of bias due to the open label trial design. I want to make a few safety remarks about the study. There were expected adverse events consistent with prior modulator trials listed here of usual frequency. There were no significant laboratory or ECG abnormalities attributed to the study drug during this study. We did have one subject who was hospitalized for a, quote, pneumonia and CF pulmonary exacerbation about 16 days into her washout period. We attributed this initially to the kind of withdrawal syndrome observed with highly effective modulators that has been reported by others in the past. This patient, however, using her data, she was unable to come for the day 56 visit to our site, but she used the data through day 28, was able to actually get off-label after she had recovered from this initial pneumonia and was able to return back to baseline with use of the therapy. Our data is remarkably consistent with other recent case series experiences in slightly less controlled environments, both from Israel and from France and their compassionate use program. And I'll just summarize this data saying that in both of these trials of a population in the Israeli study of mostly N1303K minimal function patients, in which they showed minimal to moderate sweat chloride improvements, but very large lung function and other significant improvements. And similarly, in the French study, had a larger population of N1303K homozygotes. This study showed a little bit more reduction in sweat chloride, however, very consistent, very, very positive improvements in lung function and other parameters. So our data confirms in a more controlled environment these data which were previously been published very recently as companions to this work. In addition, we have sought out to gain access for patients early with this trial data result. So this is data showing success using the individual patient's data for payer reimbursement in the US. That's a big issue for us. So you can see that 80% of patients have received off-label use of this mutation using the data we provided in specialized letters to their centers. In addition, and more importantly, 2 subjects, so fully 10% have come off of lung transplant lists as a result of this study. So really a good outcome for these patients. And finally, I want to end with saying this study has really been a model for research referrals. We have been sort of establishing these research collaboratives in the Therapeutics Development Network for some time now, and we fully engage that research collaborative network for this. So as you can see, patients came from all 4 corners of the US, and they were more than willing to come from internationally if we'd been able to allow them into the study. So really a model for research referral, and I hope we can employ that and learnings we've had from that for future studies in engaging stuff that we're talking about other of these discussions. So I want to conclude by saying this trial blends rigor with feasibility for testing ETI in rare CFTR variants. Though the sweat chloride reduction was negligible in the majority of subjects at the day 28 endpoint, we observed very significant improvements in lung function, symptoms, and weight at the day 28 time frame. And as a reminder, the regression of endpoints, the pre-treatment mean by day 56, and all these clinical parameters really suggest a drug effect and a meaningful improvement that was ETI specific. While our initial hypothesis regarding the sweat chloride reduction response to ETI were not proven, this has opened up a significant number of very interesting scientific questions. We hypothesized several things which may be going on. One is there may be variable tissue effects of the N1303 processing mechanisms between the lung epithelium and the sweat. That's a leading hypothesis for us. There may be the processing variant, although fairly analogous to F508del, is actually quite more distinct than we thought, and they may bring question to the concept of the class 2 mutations being all very similar in terms of their molecular defect. There may be secondary effects of ETI which have been observed in other clinical trials, including reduced mucus plugging, inflammation, or other effects of ETI that are off target from CFTR activation. Or finally, there may be distinct mechanisms related to transport of bicarbonate versus chloride in the sweat duct compared to the lung, which may cause insensitivity to this biomarker. If this hypothesis is proven correct in the work we're doing right now, this will be very helpful, I think, for understanding more the use of sweat chloride and other CFTR activation endpoints for rare variants like this. And so with that, I'll take any questions you may have. And thank you very much, Dr. Hill.
Marty, I think I speak for many people in the community when I say thank you for doing this.
It's our pleasure. Thank you.
It is going to make more than one person breathe easier.
Well, we know at least it's helped 18.
At least 18
yeah, exactly.
so far, and counting.
That was 16, excuse me, 16 already.
And counting, hopefully.
Yes.
I imagine there will be questions.
Yeah.
Yeah,
Felix.
Really a great data set, and happy that you did this study, be confirming the data from Israel and France that are already very puzzling in terms of the chloride sweat chloride story, and certainly tells us that sweat chloride might not be the outcome measure for everything in CF. So there might be, one of the leading hypotheses, as you said, there might be a different processing mechanism in the sweat gland and the lung. The question is that also going to be the case for other tissues, such as the liver, that we cannot easily access. Is that something that you will try to explore as well?
I think we're going to start with the sweat versus the lung, because we have model systems we can look at. The question about the other organs would be very interesting, especially for this mutation, because as some will be aware, this is one of the mutations that tends to have a lot of GI issues as well in certain patients. In fact, some of the patients enrolled had dramatic lung function improvements, but did not have significant pulmonary symptoms per se. more so than a healthy person would, but did not have, that was not their significant burden of disease. And so it would be great to understand that the model systems we've got to work out to understand that, but it's a great question.
Yeah, there's some IPC-based models for the liver that might actually work for that, so that would be interesting to explore.
That's a wonderful idea. Thank you for the suggestion.
And my second question is in terms of the reimbursement. That's a great story for what you accomplished in the US, but in terms of bringing this to reimbursement to other areas of the world, what are your thoughts on how this will help and moving this forward?
I'm going to speak as candidly as I can. I just say this data is being looked at for other ways for reimbursement, okay? So we hope that it will help with other ways for reimbursement besides just individual patients.
Thanks.
Marty, thank you for doing, this work. This is really exciting. Couple of questions. One is basically you're saying, I'm assuming that the second question that Felix asked was, mainly finding ways to approve this via the FDA. That's what you were alluding to.
Yes, we're not doing that. It was
not doing that
study. However, we hope it will help with the package to do that. Yes.
Then the other, question is a quarter of your patients were W12A2X, which is another mutation with my little knowledge that I know might have maybe some responsiveness, to this. Is there any difference between the groups of patients that you saw in any of the clinical outcomes based on what they had?
We thought that too, because we've actually published that work in our group. The W1282X, they were distributed amongst the endpoints. And so the rest of the patients look almost identical in terms of endpoints. So they were not the highest responders or any of that. They were not driving the group. So I should have shown that data, but in fact, we looked at that. No, they were not driving the boat. So yes.
Thank you.
Mike, yes, thank you.
Marty, question about liver disease. We have a 15-year-old with severe liver disease, this mutation, and ongoing drug therapy for 6 or 7 years for abscesses. And we've tried to get this approved, have been able to get it approved. Did you see liver toxicity?
None.
In this
we had no patients that had any there were modest increases in LFTs that we see with any of these modulator studies, but nothing that was self-limiting, no.
So we'll probably clearly, if we do get approved, have to do liver dosing. Do you think it'll still be effective for this mutation?
Well, Mike, I don't know. I will say that I'm speaking at a symposium this afternoon. That's one of the major topics so maybe come to that symposium. I honestly don't know. I'm not an expert on dosing of these drugs. We obviously, with standard dosing here, we did not want to rock the boat with specialized dosing for this study. There were actually a couple patients that we could have enrolled had we been able to do that with this study, but it's a great question. But I think it's one for the folks that are experts in dosing and PK and whatnot to answer.
All right, thank you.
Yeah, sure. Great. Thank you all very much.
I have a comment and a question. So I take care of a young man in his mid-20s. So besides his CF, he also has severe cerebral palsy and significant developmental delay due to that. So therefore, he doesn't do PFTs. So we go by how much he's coughing from what his father hears and how much mucus is kind of sitting in the back of his throat. But his family was so committed, they paid a month of ETI out of pocket, $28,000. And then we did sweat before and after and saw no response, and therefore he has an N1303K in a minimal function. But again, thank you for this data, because now there is a reason to go back and try. His father swore he did better on the drug, but we had no way to measure because we had no PFTs.
Stabilize it could help, right?
Generally very healthy at this point in his mid-20s is still healthy. But anyway, thank you for that. It's clearly going to have far-reaching impact.
And our goal is to rapidly try to publish this data as quick as possible to get it out there for the community. And there are, as I said, the Israeli study and have very recently been published, but this is a larger data set with a little bit more control, so it may help with the approvals. So.
Marty, there's one question in the chat.
A few questions.
We've got a couple here. Let's try not to hit this one too far out of the park. What does the N1303 data tell us about the limitations of sweat tests and the HRT system in CFTR modulator research?
Yeah, I think it's an FRT system. I'm going to say the FRT system that's employed by Vertex has been extremely beneficial, right? This study was in no way meant to throw any sort of shade to that system, rather to just point out there may be particular variants that are less understood that we cannot use one system to understand by. That's strictly the point of this, and it's strictly be a translational study. I think the sweat point, the sweat is a wonderful group biomarker. We've all had publications looking at that, but when you think about individuals, breaks down, and there's minimal data on significant level correlations with individual sweat. So I think there's a few problems with the sweat. It is a great thing to tell us, hey, you're activating CFTR, but as we know, that is not the whole story, and so we certainly have to learn more. I think if we are able to accomplish understanding some of these hypotheses we put forward, that may answer that question better. But at present, I think it just tells us that in these rare variants, we have to learn more about how they work in the sweat gland. And so it's probably that issue of biology rather than being a problem with the gland, with the assay itself. And Edith can comment on that more after for others that have. I'll defer to her for questions about that. So I think we've had those types of discussions before. So yes.
There's another question here. I think sort of referencing an anecdote perhaps of a local patient. So it says they had a local patient who had a classic purge. So do you have, I guess, did you see that, and do you have any thoughts on sort of measure of sputum volume, quantity, composition?
We didn't bring that in because that had not been sort of a validated endpoint for the studies. However, I would say I'm gonna guesstimate that probably 2/3 of the patient had some level of a classic purge, the kind of 3 to 7 days after, coughed up a lot of mucus and then felt much clearer, even if they weren't the heaviest mucus producers to start with. So many of them had that purge, but that was strictly just anecdotal, no endpoint purpose for the study.
Okay. I have one just to wrap up. So you didn't have any N1303 homozygous patients, I don't think, in the study. Do you think there'd be a gene dosage thing if you did have 2 copies?
French data suggests that of their 8, I think 6 or 7 were homozygotes. So they had a chloride reduction of almost 20. So would argue that's the case, but I think it's hard to say that the studies were done somewhat differently and whatnot, in the environment. So I think it's hard to say for sure. We hypothesize there might be because, but again, it's hard to say for sure. We don't have we don't have any N1303 homozygotes. We're hoping to have a few in this study to answer that very question with the in vitro assays. We don't have that. We didn't enroll any. So which we could get in a rare population. So great. Super.
Thank you so much.
Thank you all again.
And our next speaker is Dr. Ratchin from Hospital for Sick Children in Toronto, Canada.
Well, thank you for having me. It's a bit difficult to follow this exciting talk, but I'll do my best, and maybe. and maybe some of the outcome measures that I'm going to talk about might be helpful for some of the more complicated patients, like the one you mentioned. If I can ever get this started, that would be wonderful. Oh, yeah, sorry, that's what I did wrong. Should know by now. Okay. Here we go. Okay. Thank you. So I will talk about hyperpolarized imaging for new therapies in cystic fibrosis. This is a study that was kind of the first effort to do this in a multi-center setting within centers that have utilized this new imaging technique. And I'm going to show you some of the data from this trial. But before I do so, these are my conflict of interest. These conflict of interest slides tend to be up for about 5 seconds, so I'm doing it a little bit longer. So one of the outcome measures that we used within this study is also MBW LCI. And I'm going to focus in some of the data later on also on in terms of what we learned in terms of the cross-correlation between some of the outcome measures. So just as a background, so hyperpolarized xenon MRI, and you may know that previously helium was also used as an outcome measure, but it's moving now more towards xenon as the tracer gas to be used. It can be combined with structural imaging and ultra-short echo time MRI. And that's the advantage of that, and the nicety of that is that it can provide both structural and functional information. And there are some data suggesting that this might be a sensitive outcome measure to capture and track, especially early lung disease. And there are some data that from the UK suggesting that it might actually pick up more in terms of disease progression than other outcome measures, which is going to be interesting in the setting of ETI, where we don't know how much disease progression is going to happen, if any, in patients that are on these therapies. So the hyperpolarized imaging study for new therapies, or HighPoint study, had kind of three key objectives. The first one was to establish the inter-subject repeatability and reproducibility of the outcomes within a multi-center setting. There was some data previously obtained within a single-center setting, but we felt it was important to do this in a multi-center setting, and to determine the change from baseline to day 28 for xenon MRI ventilation heterogeneity, which is the main outcome measure that's usually used as kind of the black areas that you see after inhalation. This is where the ventilation doesn't go, and you can quantify that and ventilation defect percentage after initiation of ETI in a multi-center study. And then finally, and I will focus on that a little bit in my talk, to determine the relationship between xenon MRI and established lung function measures such as spirometry and multiple breath washout. And you can argue whether multiple breath washout is established, but at least it's been used more frequently over the last couple of years. This is kind of the study oversight. We are kind of more located to the east of North America. The principal investigator of the study is actually Jason Wood from Cincinnati. I'm the co-PI for this study. Kelly Thornton is the project manager. And in each of these study centers, and I think that's important for this for this outcome measure, we had a nice combination of having image experts and a clinical liaison. And you see the leads for the centers, and then we had the lead clinical research investigators at each of the sites, with Aviva being the one from Toronto who's here in the room. So you can ask her any questions afterwards. So this is kind of the study design overall. It looks a bit complicated. So there were two phases of the study. The first one was to look at the variability part. Patients could enter both parts of the study, but the patient population is not necessarily identical. So in the first part, it was basically a visit where two measurements were done on the same day, and then a follow-up visit after 28 days to repeat the measurements. And the measurements that were done focused on imaging, and both on hyperpolarized gas imaging and the structural imaging. I'm not going to show any data about the structural imaging today, even though those correlations will also be quite interesting, and we'll look at this into the future. And then the second phase were capturing patients before they go on ETI, and then have them seen after 28 days, 6 months, and 12 months. And some of the later time points are still ongoing as part of this ongoing studies. We had a bit of a delay in terms of ETI coming to the clinic in Canada versus the US, which seemed to be a disadvantage in the beginning, but ended up being actually an advantage, because many of the patients in the US went on the drug very quickly, so getting a pre-measurement was not so easy. So some data for the first part of the study. Again, this is a visit where two measurements of xenon MRI were done on the same day, and then a follow-up visit after 28 days. This is I'm showing you the baseline characteristics of all participants. So 38 patients participated in this study, and this is the breakdown by sites. I didn't name the sites in this slide. So the age group, this focused on pediatric patients. So this is a study on pediatric patients, and also focused on patients with relatively preserved lung function. And you can see the mean FE1 is in the 100 to 100 plus range, and it's also shown are the LCI data and the VDP data at the baseline visit, which varied a little bit between sites, but not very dramatically. And when we look at the same day reproducibility, so this is shown as the difference between the scans, like versus the mean, kind of a blunt Altman plot. And there are two different ways like this was being looked at. One was the classical VDP measurements, and one was another measure that has been developed by the group in Cincinnati. I'm not going to go so much into the details between the two methods, but overall the difference between the two scans was very small, and except for one outlier, which is shown for the RDV method on the right-hand side. There was quite close correlation between those measurements done on the same day, and it didn't seem to differ much between patients with different levels of VDP. If you look at the between-visit reproducibility, so this is between visit 1 and visit 2, there was no significant change in VDP between these 2 visits. There was a bit of a change in FEV1, which was somewhat surprising, and no change in LCI. And I will come back to some of this correlation between these outcome measures a bit later, and we'll show you that the correlation between LCI and the VDP measurements was actually quite close. And these are just examples of 3 images of patients done on the same day on visit 1 and on visit 2, and the patient 1 and 3 with very stable measurements over time, and in patient number 2, you can see some changes on the initial image that was done in the first scan, which then changed to the second scan. So it was, well, overall the message was that the reproducibility was quite high. It wasn't the case for every patient, so there's some variability in the measure. But let's focus more on the phase 2 data, because the phase 1 data have already been presented before. Again, the major objective was to measure the change in xenon MRI ventilation defect percentage and in patients after initiation of triple combination therapy. And then we wanted to correlate this to other measures such as FEV1, but also LCI. But the primary endpoint was really the change in VDP over time. I'm going to show you the distribution about the enrolled patients, and if we wanted to get 24 patients in the study, we will probably end up with 26 patients if everybody ends in the study, but at least we will meet the goal of 22. You can see there was a bit of a distribution between the different centers, and again, that was caused by the fact that after approval, the uptake of ETI was very rapid in the US, so we had to jump in and compensate for that in Toronto, which we did. For the group overall, these are the changes that we saw. Not surprisingly, we saw a significant improvement in FEV1, which you would expect. There was also a reduction in LCI, as supported by previous studies. And then we see this reduction in xenon MRI VDP, and all of these are highly significant, and which to me is not very surprising, because this is a highly effective intervention. So this was probably not the most interesting part of the study, even though it was an important component to show. And just to give you an example of this, is that this is a patient where we see a response in all of these outcome measures after initiation of ETI. You can see the baseline imaging, and then after 1 month after introduction of ETI, the VDP is reduced, the FEV1 is improved, and the LCI has been going down. So how does that relate to correlation between these different outcome measures? And I show you 2 different ways of looking at correlations. One is the correlation between the outcome measures at baseline, and then after in terms of the treatment effect. So this is the baseline correlation between LCI and VDP on the left-hand side, and between FEV1 and VDP on the right-hand side. Both are correlated, one positively, as you would expect, and one negatively. But you could see that the correlation is much stronger for LCI, which is not surprising to me, because LCI is supposed to pick up ventilation in homogeneity. And so that is reflected in that. There's a correlation with FEV1, but you can see there's considerably more noise. So how does that relate to treatment effects? This is here shown on the next slide. So this is the absolute change in LCI plotted against the absolute change in VDP on the left-hand side. and the absolute change in FEV1 to the absolute change in VDP on the right-hand side. You can see both are reasonably well correlated, but again, we see a stronger correlation between LCI and VDP than we do for FEV1. And that really raises the question of how much additional information do we get from the imaging versus the LCI methodology, and this is something that we have to look more into as we move forward, because certainly the imaging technology is a bit more high-end and more difficult to do in a multi-center setting. But overall, I think the data are promising in terms of showing that you can actually do this in a multi-center setting. So this is where I come up with my conclusions. So the hyperpolarized xenon MRI is feasible in a multi-center setting in pediatric people with cystic fibrosis. The results overall are reproducible, both within but also between visits. As you might expect, the VDP improves after initiation and ETI, but the response is not uniform, and there are some patients where there's a discrepancy between the imaging and the FEV1 outcome, and we've seen this if we look at individual patients. Again, it's more consistent if you look at LCI and versus VDP, and this kind of tells the story that was also brought up yesterday in one of the sessions, is that you may see different in outcome in responses to the different outcome measures, which might help us to understand a little bit better whether a patient actually does respond or not. So simply not responding in one outcome measure may not tell the whole story. And so both baseline measures as well as treatment responses are strongly correlated to LCI. It's less so for FEV1. But the longer-term data will really help us to elucidate the role of hyperpolarized gas MRI in tracking disease, because what we really want to know is there stability with that over time in patients treated with ETI, or do we see some worsening over time, which would be very interesting for us to clarify whether this 1-year study might not be enough to pick this up, but at least it will give us a first glance of whether that's the case or not. And with that, I like to thank people that have been involved in this. A special thanks goes to all the participating children and families. These are not easy studies to do, but the patients and families have been very engaged in helping us to understand this outcome measure better. And also the clinical teams to facilitate integrated research visits, because that's also sometimes challenging to do this in a busy clinic. And last not least, the CFF for funding this study. And with that, I'll stop and I'll be happy to take some questions.
Thank you so much. That's, again, the images are striking, and really finding a place this is both a clinical and research tool will be interesting to see in the future. So I just wanted to ask, and I think I may know because I believe I serve in the DMC for your trial, but maybe for the benefit of the audience, safety of inhaled xenon in CF patients and issues, especially since it seems most useful in the younger population with mild lung disease, issues with performing the imaging and claustrophobia.
Yeah, thank you. Those are important points. In terms of the xenon safety, we haven't seen any significant safety signals. So the xenon inhalation is somewhat low in oxygen concentration. So there can be some transient dips in the oxygen saturation if you inhale this. We have not seen any prolonged issues with that. But this is also a population of patients with relatively mild disease, so this might be more of an issue in more advanced diseases. So there are now efforts underway to do this in younger children. I mean, and in Cincinnati, they're also using this to study infants. So and so far, the safety profile looks very promising. The claustrophobia is certainly an interesting component, and it may not be the ideal technology to be used in young children, and there are other imaging technologies that do not necessarily require hyperpolarized gases that might be more amenable for that. And one of the nicety of this data set is that we can actually cross-validate some of these other imaging technologies with the hyperpolarized gas imaging, which is still considered kind of the, I shouldn't say gold standard, but somewhat standard in the field. And so this is an opportunity to look at that, because it certainly will be interesting to also move into these younger age groups. But keeping young children happy in an MRI is not easy. So.
Andy,
just to follow on, I'm sorry if I missed it. How long does this protocol actually take? How long are you actually in the scanner, and is this practical for young children, which you've passed down?
Yeah, it depends a little bit on what you tend to do. If you just do this one measurement versus if you add on some of the other structural imaging. So the actual imaging also depends on whether you do one dose versus two dose. So, I mean, the actual time in the scanner is less the acquisition of this image versus all the time around it. So which takes about half an hour to an hour. But really getting somebody stable in the scanner is more of the challenge. And this is where it becomes more difficult in
basically, they're in a scanner for 30 or 60 minutes ballpark.
So, yeah.
well,
Well, because we do some extra stuff.
They get a little break once you switch over to the proton.
Yeah. Okay.
Hi, Shijing Jia from University of Michigan. Can you comment generally on whether there's any utility in different patterns of ventilation defects, or is the utility of this clinically and for research more the overall percentage? As in, for example, is there a distinction between small airways disease versus large airways disease, migrations of patterns that might be able to distinguish between perhaps CF and asthma?
That's an excellent question. And I think what we currently do with these imaging technologies is kind of the poor man version of analysis, because we basically look at these black areas and say, okay, these are more black and these are not so black. One way to look at this is in histograms in terms of the distribution of that, which might be a bit more sensitive. But I think this is still a lot of this is still a work in progress in terms of how we can better quantify the large amount of information that you get, and also in terms of looking at the variability of response. When this was first done with hyperpolarized helium in patients on ivacaftor, one of the reasons it took so long to get this manuscript published is because the investigators tried to make sense of the different responses that you see on imaging that you cannot really capture with other methods, and because the ventilation defects look very different from patient to patient. And how do we deal with that, and how we utilize that information in terms of the clinical correlation is still something that we have to learn. And I should probably say that there are other studies going on doing this, including my co-chair, the co-chair here, Jonathan, who's doing some studies with that in Vancouver as well. And there's going to be some presentation or a poster on that, right? So there's a poster presentation at the same time.
I think it's happening right now. Yeah. I have a question for you, Felix. The trade-offs between sensitivity and variability in different outcome measures, so sort of FEV1 versus LCI versus VDP. I think the retest component of this study was maybe not as sexy, but actually super important. What did you learn from that about the MCID of VDP, and are the changes that you were seeing relevant, important outside of the wobble of stochastic mucus bouncing around in the lungs in people with CF?
Yeah, that's a very important question, and we are actually not quite at the point where we have analyzed that adequately. So one of the analysis that we wanted to do is to also look at variability in terms of predicting the response. And Sanja Stanjanovic is much more skilled in her statistical analytical capacity than me will hopefully help us with that, because that's also something that we might learn, that the variability is not necessarily noise but actually signal. And we've seen this in some of the LCI data, that initially we thought, oh wow, this is quite variable, but then it's actually related to signal if you have a sensitive test, as you alluded to. So this is something that we need to understand better, and it's not very simple to sort out. But this data set will somewhat help us, because at least we have these short-term measures. And it's also interesting that we see, and you're involved in this as well, when we do some studies in other diseases like PCD, the variability seems to be higher because the mucus tends to move around all the time. So that those are also things that we need to learn about, what is CF specific and what is not.
Any more? One more check online.
Oh, yeah, I think you, with 40 seconds left, I think you mentioned it, but there's a question just about whether sedation is required.
Oh, no, these were studies that were not done under sedation, so we don't think that's a very feasible test if you need sedation on a regular basis, especially if you want to do this repeatedly. How this will translate into studies in younger children, that's still a bit of an open question, but I think it's important that we establish this technology in older subjects where you can do it without sedation, and then think about moving it to younger age groups.
Thank you.
Super. And our next speaker, continuing along with the observational study theme, is Raksha Jain from UT Southwestern, talking about the Mayflower study, which is also very exciting. It's one hit after another.
All right, well, thank you to the moderators for inviting me to present these data on behalf of the entire Mayflower study team. So this study, prospectively evaluating maternal and fetal outcomes in the era of CFTR modulators, is being sponsored by the CF Foundation and run by the TDN, and it's really taken a village to get this study going, but we're very excited to share some interim results with you today. These are my disclosures. So just to give you a very brief background, I think we all know as people with CF are living longer and healthier lives, many more are pursuing pregnancy these days. If you look at this graph on the right-hand side, this is data taken from the CF Foundation registry, where as you see survival, median predicted survival in people here in the United States in particular improving, you see that the number of pregnancies in this yellow line really tracks along with that. In addition, the peak that you see over on the right-hand side really occurred in 2020, 2021, and 2022 after the release of highly effective modulators. There's been a number of studies looking at pregnancy in CF, but many of these have been retrospective, they've been single-center studies, and they've been done in times prior to the release of highly effective modulators. So the goal of this study was really to do a prospective observational study in a multi-center fashion to help us as clinicians guide care for our patients who are interested in pursuing pregnancy. So the primary aim of this study is to assess whether changes in FEV1 surrounding pregnancy differ in people with CF and how this impacts their FEV1, and particularly looking at whether that change in FEV1 has any difference in those who choose to stay on modulators versus those that don't. We'll also have the benefit of being able to factor in a number of comorbidities in our large sample size to look at how this may also influence FEV1. There'll be a number of secondary outcomes, including describing pregnancy-related and obstetrical complications, as well as changes in mental health that are being captured throughout this study. And there's also a number of exploratory aims, including sub-studies that are going to evaluate CFTR modulator pharmacokinetics in mother and infants, and a sub-study being led by Melissa Putnam on looking at glucose controls surrounding pregnancy, as well as infant outcomes. So showing you a little bit about the study design and methods. First, it's important to note that this was an observational only study. We're not intervening on any aspect of care that the clinician and patient choose to pursue as part of their pregnancy. We're simply capturing very granular data about the pregnancies and particularly about the infants, because I think that has been one piece that has been missing dramatically in the literature is what do we know about infants born to mothers with CF? But this is a 41-site observational study being conducted here in the U.S. The aim is to enroll about 285 people with CF, and the study began in September of 2021. The eligibility includes people who are at least 16 years of age, currently pregnant, and planning to continue their pregnancy. So showing you a little bit about the study visits, when a person is pregnant, they're eligible for the study or confirmed to be pregnant. We attempt to capture a visit in first trimester, second trimester, and third trimester. If we don't capture the earlier visits, that's okay. They're eligible at any point during their pregnancy. And at those visits, we're capturing lung function, we're capturing some questionnaire data, we're capturing mental health scores, and we're capturing a blood sample in these people. Then at delivery, there's no distinct visit in the majority of the core of this study. And postpartum, we're following these patients every 3 months for the first year and every 6 months for the following year. And very importantly, we're following that infant data also for up to 2 years post-delivery. The continuous glucose monitoring sub-study I mentioned that's being led by Melissa Putnam is also very important, as we know very little about glucose control in pregnant women with CF. And this study is aimed at capturing a subgroup of people within the study at 4 visits during their pregnancy, ideally before 20 weeks of gestation. And these people will wear a continuous glucose monitor for up to 10 days at these 4 different points during their pregnancy, so we can really get information about what their glucose metabolism is doing throughout pregnancy. And these can be people who have CFRD, people who don't have CFRD, people with impaired glucose control, actually all comers, so that we can really understand it at all levels. And then after delivery, we're seeking another visit where they wear another CGM 8 to 12 weeks after delivery. There's also an ETI pharmacokinetic sub-study that's in collaboration with Chuck Esters at a UNC, where in this study, this is again a subgroup that we're capturing here. There's 2 cohorts. One cohort is going to be collecting specimens at the time of delivery in mom, in cord blood, and in the infant. And we're going to get very careful information about when the last dose of ETI was surrounding that delivery. And then ideally a visit that is done within the first 6 weeks after delivery. And again, capturing, and those are in moms who are breastfeeding, to capture maternal blood, breast milk, and infant blood. There's also a second cohort for those people who may not be delivering at the site where their study is being conducted, and that is in those that are choosing to breastfeed. We capture similar maternal blood, breast milk, infant blood at 3 months, and ideally at 6 and 9 months in both of these cohorts if the mother is still breastfeeding at that point. So again, these are interim results, so we haven't done statistical analyses on these, but I'm just going to share with you where we are as of September 1st, 2023. So we've enrolled 206 people in the core part of this study, so big thanks to all the sites that have been participating. In the CGM sub-study, we have 38 enrolled right now, and in the pharmacokinetic study, we have 25 consented and 12 officially enrolled in that pharmacokinetic sub-study. So sharing a little bit about these 206 people and what we know thus far. The mean age of the population is 29.8, but you can see there's some as young as 17 years of age and some as old as 41. The race is as you would expect within the CF population. The large majority are white, but we do have other races represented. When it comes to ethnicity, 94% of the population is non-Hispanic. We do have almost 6% that are Hispanic in ethnicity. The genotype is largely 50% F508 homozygous, 30 or so percent that's heterozygous, and then others as well. Diabetes is actually more prevalent in this population than I would have expected, but we have about 36% that have CFRD and 2 that are type 1 diabetics. The baseline BMI in this population, fairly healthy at 24. And when you look at our baseline lung function, this is a fairly healthy population where the mean FEV1 in this population is 82. But importantly, when you look at the range, we have people as low as 28% FEV1 and as high as 120. So this will really give us an opportunity to look at the spread of FEV1 and how this might impact outcomes in both mothers and babies. When it comes to their pregnancy history, this is what we have thus far. When it comes to who used fertility assistance, about 12.7 or 26 people have used fertility assistance thus far in our study. Those who are actually having the father tested to be CF carriers are the majority, which is good news. So about 72% or so are getting the father tested. In our study population, 10 fathers have been identified as carriers thus far. And when it comes to previous pregnancies, the majority of these patients actually have had previous pregnancies in the past. So this is our spread of percent predicted FEV1 over time. And very importantly, this is not statistically analyzed. And I do want to point out that this is separated in time by baseline, the 3 trimesters of pregnancy, and what we have post-pregnancy thus far. But if you look at the post-pregnancy data, we're still waiting for people to reach those time points. So the population decreases over time, or that last time point at 7 to 9 months currently is only about 30 people. So take that with a grain of salt. But if you just look at the trend, at least you can see during pregnancy, there's fairly stable FEV1. Early in post-delivery, with those numbers that we have so far, there's a subtle dip, and it's really hard to make conclusions beyond that, knowing that we don't have our full population set yet. Looking at weight over time, it's as would be expected, where there's a nice weight gain that these mothers have experienced during their pregnancy. And actually looking at the data we have thus far post-pregnancy, they really return to about baseline weight post-pregnancy in the majority of them. So that's good to see thus far. From our CFQR respiratory domain data thus far, you see that overall in pregnancy, there is a slight decline that we see with a little nadir thus far in the 3rd trimester that recovers post-delivery, and there could be lots of explanations for that, but again, remains to be seen what we see after this, and maybe is not too surprising when people feel terrible in their 3rd trimester pregnancy. And then you look at mental health scores, and I think this will be a really unique aspect of this study where we're actually capturing these as part of this study in a really methodical way, where we have PHQ-8 and GAD-7 scores in this population. The good news overall is when you at least look at the spread of this, we're seeing overall that the numbers are low throughout pregnancy when it comes to both depression and anxiety. But of course, there are some that are high, and I think we'll need to take a bigger dive into that later on. Who are these people? Why? And is this changing in relation to pregnancy in any way? And again, break this down by those who are on modulators versus those who are not on modulators. So when it comes to delivery outcomes that we have thus far, there have been 102 live births that are full term in our cohort. There have been 28 live births that were premature. There's been no stillbirths, 6 spontaneous miscarriages or abortions, and 4 induced abortions. When it comes to modulator use during pregnancy, the majority, actually the large majority, are using modulators, not surprisingly, during their pregnancy, but we are capturing those who stop at different points during their pregnancy, so we can factor that into the analysis at the end. And when you look at the proportion of time spent on modulators throughout pregnancy, it is again the large majority of the pregnancy that people are continuing. And that's probably not surprising to most of us, I think. So as far as infants' characteristics for live births goes, when we look at APGAR scores at 5 minutes, they're fairly good overall with a mean of 8.75. Gestational age overall is 37. The weight of these babies is fairly good. There are some low weight babies that are being born, but the mean is 3 as currently, length 48, and occipital frontal circumference 34. So overall, I think the mean is looking like we're seeing healthy babies in this population. From a delivery complication standpoint, the majority of these babies are being born vaginally. There are about, I would say when you look at the classic C-section and low transverse C-section population together, that adds up to about 30% that are undergoing C-sections. And that is fairly similar, I think, to the general population. And then when you look at infant complications, I've just highlighted some of the ones that we see in greater than 15% of these babies, and that is extended hospitalizations or NICU stays, which are at about 20% in both of those. Jaundice at about 15% or so, and preterm labor, 22%, and respiratory distress syndrome, 12.4%. So this is not significantly different than what we've seen in CF reports before, but this is only 113 of the babies, so we have a lot more to learn, but we do need to understand a little bit more about potentially the risks of who are those babies that are experiencing some of these more adverse events. And I think another really nice part of the study is we're capturing infant milestones or baby milestones 2 years after birth. This has been something that's never really been done before, and I think many of us who are enthusiastic about keeping our mothers on Trikafta throughout their pregnancy, I know I'm receiving questions for them, not just about the birth, but about the implications, particularly on their mental state after birth, and so this should allow us to look at this more fully. With the data we have right now at 3 months and 6 months, we see that if you just want to focus on the 75% milestone, which I think a lot of people think in, that overall about 89% of babies at 3 months are reaching that 75% milestone, and 83 at 6 months are reaching that. So good news overall, I think, and hopefully that can make us feel a little better. But, again, I think we want to take a look at those who don't and understand whether there's something that puts babies at more risk. So just to give you some preliminary conclusions, and again, we have not statistically analyzed any of this data. It's only interim analysis alone with the patients we have thus far. But the majority of these people are continuing their CFTR modulators for all or part of their pregnancy. It's really too early to draw any definitive conclusions about the mothers or the infants in terms of outcomes. But the trends we're seeing are that the percent predicted FEV1 is maintained during pregnancy. There's an early signal that there may be a decline early postpartum, but more to see on the recovery of that and what it looks like when the rest are enrolled in this study. That the weight is increasing appropriately during pregnancy and maintaining. The CFQR, really, we saw the biggest decline in third trimester. The PHQ-8 and GAD-7 scores are fairly low throughout. And the premature births are a little bit higher and spontaneous abortion rates a little lower than the general U.S. population. But overall, the infant data we're seeing are fairly similar to what's been reported in the CF infant population in the past. So with that, I really want to thank the people with CF and their families who've been willing to participate, and thank all the sites that have put a lot of hard work into this study, and particularly the Mayflowers team and my co-PI, Dr. Jennifer Taylor-Kauser, on this study as well. Thank you very much.
Okay. All right. Do we have any questions from the floor? Felix can ask. I'll start before he goes. So there were 10 fathers who were carriers. Mm-hmm. So my quick back of napkin tells me that there should have been 5 babies with CF.
Yeah, so we looked at that. We thought that would come up. So far, we do not have any babies with CF reported. Now, I mean, we're waiting to find out the full knowledge of all this, but right now we do not have any babies with CF reported. I will tell you there is a session this afternoon, a pregnancy and fertility symposium. I think it's symposium 18, where Chris Fortner is going to talk a little bit about his experience in taking care of infants who've been exposed to CFTR modulators. But we know that's a huge question. The data is what the data is, and we have not yet seen one in our.
Do you have any sort of a priori plans for what happens, like, or any additional tests for any babies with CF who are born and moms are breastfeeding and on ETI?
We didn't build in anything into the study, so it's not part of our consent process right now. And right now, all we're going to do is, if they enroll in the PK study, that will be terrific. That would take that baby being born in a site that's participating in the PK study and that mother delivering at that site. So there's been lots of challenges with that. But we hope to have that data. And if the mother chooses to breastfeed in addition, that would be really nice to have. But we'll have all of that infant birth information. We'll have the pediatrician visits afterwards. We've received consent to get all of that information on these babies. So I think we'll have a lot of information, but how many babies this is going to be and what we'll see, we have no idea.
If they happen to have a sweat test done, you can maybe use that too.
And we will be able to collect the sweat test if one was done. That's correct.
great talk. Do you have any information about breastfeeding rates in this population? Because
I do not think we have yet reported that data. That's a good question. I don't think we've captured that yet. We still have a lot of data slowly coming in, but I don't think we have it yet. So
yeah, it would be interesting to know what maternal choices are in that setting. Yeah.
Yes, absolutely.
Thanks for your talk. Really interesting information. I was just wondering if you saw any infants specifically with electrolyte abnormalities and metabolic issues that were admitted to an ICU after birth. I'm speaking because we have one patient whose mother was on ETI who was admitted to our PICU with metabolic disturbances, and difficult to know whether that was ETI related or something else. The patient hasn't shown for follow-up, so we aren't able to really discern whether or not they do have a positive sweat chloride, but they didn't initially. They were pretty low.
That's a good question. We don't yet. We have all of this information, and we've tried to make it as easy for the sites as possible by bulleting different categories, but there is room for them to add complications that are not on the list, for example, that I have here. So if they find an electrolyte disturbance, they could enter that in, but it's not something we specifically captured as a bullet point in this to have that data so clearly
this was
available.
not subtle. The kid was in the ICU, but interesting to me. Thanks.
Hi, very interesting study. Two questions. One, I don't know if you told and I missed it. Are you planning to reach a specific number of people enrolled in this study? And the second question is regarding the exploratory PK study. What kind of samples and measures are you planning to do with these samples?
Okay, good question. So we're planning to enroll close to 300 people in this study. Now, we may choose, particularly for these sub-studies, to over-enroll a little bit to make sure that those sub-studies have a robust population of people that are captured. When it comes to that pharmacokinetic sub-study, we're largely capturing blood samples, but that's maternal blood samples. At birth, we're also capturing cord blood samples and infant blood sample. Then for those mothers that are breastfeeding, we're getting mother's blood sample, breast milk, and infant blood samples. And Chuck Esters at UNC is going to analyze the pharmacokinetics. We're also going to analyze liver function tests if able to do that as well.
Thank you.
Thanks so much for representing the study so well. To answer your question, the CF Foundation worked with us to actually create a guidance document so that anybody who's on ETI during pregnancy, we recommend that they get sweat testing and genetic testing on their child. Of course, sometimes the fathers are tested, but they may not actually be the father. There are lots of reasons why just doing the fecal elastase and all of that won't work, because you need to know what actually happened to the baby. So that's a recommendation that's out there for all the people in the study, but just across the US. Like anybody who's on ETI should get genetic testing and sweat testing on their baby.
Right. Yeah. I mean, I guess if 28% were unknown sort of paternal genotypes too, right? So there will be a few more than the 10.
Right. Yes. And then the only other point that I was going to make regarding the outcomes in the infants is that our population is older than the average population in the U.S., and there are a number that are considered advanced maternal age. For all the pulmonologists who don't think about pregnancy, that's 35. So we do have an older population who are getting pregnant. So we have to keep that in mind too, and we will as we analyze our data on the infant outcomes.
Thanks, Jen.
Thank you for this. Thank you for the great study. I had a question for the infants. Is there any plan to seek eye exams from their in utero prolonged exposure?
Yeah, so that's a great question. And there have been some reports of infants born with cataracts who've been exposed to Trikafta in utero or while breastfeeding. And thank you for that question. The key about this study is we didn't want to mandate anything. We wanted to make it observational only, where the decisions from the moms and about them and their families were coming from their clinician and their center. So we're only capturing data. I think, as Jen mentioned, that there is this guidance document, and a few of the things we are suggesting in this guidance document is also for those that continue ETI to have the infant checked for liver function tests, to have eye exams done, but we're not mandating that as part of the study. If they get it, we are absolutely going to capture that because we're very curious, but we didn't make it part of the protocol to insist anyone do any of these tests.
And we're down to the last 57 seconds, so I think there's time for one more question.
Hurry up. Thank you so much, Raksha and Jennifer, for doing this study. I have a very quick question, and that is, are you capturing help with childcare after delivery and somehow connecting it to maternal health outcomes and infant outcomes?
Yeah, that's a great question. Yes, we are. We are capturing how the baby is going to be taken care of. And we're capturing a number of other things after delivery as well that I really didn't get into today. But our goal, including whether they're started on contraception soon after pregnancy, all these different things that we want to look at. So that's the real advantage, is we're going to have a huge data set. And, we welcome anyone who has interest in looking at this data. As with many TDN studies, we want people to be able to utilize the data for additional questions we may not capture, but that is part of what we're going to be collecting. So thank you.
Thank you so much.
Thank you so much.
Thank you. I'll just throw in one last comment for that. As a participating site, I echo your sentiments that the number of patients that were willing to step up and participate. This is a stressful, vulnerable time, and they were all majority very eager to participate because the outcomes will help them and those they love. All right, and our last talk will be Dr. Urania Rappo from Biome X, who'll be speaking to us about phage therapy.
Click on yourself and then click start. Yeah, there you go. Because you wait a little time anyway.
Thank you, everyone, and thank you to the moderators for inviting me. I will be speaking about a phase 1b/2a randomized double-blind placebo-controlled multicenter study evaluating nebulized phage therapy in CF patients with chronic Pseudomonas aeruginosa pulmonary infection. I'm at Biomics, and I'm a senior director of clinical development, also the medical monitor on the study, and an infectious disease physician. These are our disclosures. Biomics is a publicly traded company, so I have the slide up. So I will present a little bit of the background on how CF has still an unmet need in Pseudomonas aeruginosa infections and how phage could be a potential solution. And I will speak about the phase 1b/2a randomized clinical trial of nebulized phage for patients with Pseudomonas. I'll go over the objectives, the methods, the top-line results from Part 1, and the Part 2 demographics in the blinded part of the study, which is still ongoing, and a conclusion. So most of you know here, if not all of you, that Pseudomonas aeruginosa is the most prevalent pathogen in cystic fibrosis patients, and at least 50% of CF patients are infected. This is a highly problematic pathogen, and the colonization is associated with lower FEV1 at all ages, as shown on the right graph here, and long-term morbidity. And after prolonged and repeated antibiotic courses, the patients have increased antibiotic resistance and lower efficacy. So it becomes a worse problem as patients get repeated infections with Pseudomonas. And more interestingly, most patients remain infected with Pseudomonas even after treatment with CFTR modulators, including ETI. So phage could be a potential solution here, and many of you know the bacteriophage therapy is targeted. It binds only to targets of bacterial organisms that the phage has been proven in vitro studies to have activity against. So it does not bind to human cells, and it does not bind to other strains of the same type of bacteria, so not other strains of Pseudomonas other than the ones that it has activity against. And it is self-amplifying, and it is a novel alternative or adjunct to antibiotics in chronic Pseudomonas aeruginosa infections, and has been successfully used in compassionate use cases in pulmonary infections, including in CF patients. And here you'll see the phage life cycle with Part 1, how the phage actually locate susceptible bacteria that are targets, how they inject their genetic material, infect the bacteria, then multiply within the bacteria, assemble their virions, eradicate that bacteria that was infected, and then seek other bacteria. BX04A is a phage cocktail that targets Pseudomonas aeruginosa in chronic pulmonary infections in CF. and it is nebulized. And here I have the 3 phages that are in our cocktail, a little schematic that is, and the nebulizer that is used in our study. So it's a handheld nebulizer, and the patients open a vial of each of the phages and drop the contents of the vial into the nebulizer and then inhale a dose. The key features of BX04A is that the cocktail is potentially effective against antibiotic-resistant strains, enables biofilm degradation, and it has a potential impact on Pseudomonas levels and improved clinical respiratory outcomes. The objectives of this clinical trial are shown here. The primary objective is a first-in-human trial or safety and tolerability of BX04A, and we have exploratory objectives of the effect of the phage cocktail and Pseudomonas burden in the sputum, the PK of the BX04A phage in the sputum, and also clinical outcomes. It's a multi-center study evaluating this therapy in outpatient adult CF patients with sites in the US, in EU, and Israel. So I see many familiar faces from our sites here. Thank you for coming. And the sites in EU are sites in Spain, Czech Republic, and Netherlands, and we have 5 sites in Israel. And I want to give a special shout out to our colleagues in Israel who were not able to come today. The key inclusion criteria are an FEV1 of at least 40% in patients that also have chronic Pseudomonas aeruginosa pulmonary infection. They have to be on inhaled antibiotics. So we're giving phage on top of inhaled antibiotic therapy. And the 3 antibiotics we allow to be concurrent with the study drug are tobramycin, aztreonam, and colistin, one of the 3. And we test all the Pseudomonas morphotypes and make sure that they're susceptible to our phage cocktail. And the way we determine that is in vitro, through testing the actual isolates of the patients from their sputum and seeing if the isolates are susceptible to at least one of the phages in the cocktail. We have safety follow-up until 6 months after the last dose, and we also sequence the Pseudomonas strains by next-generation sequencing. So the study is split into 2 parts. Part 1 is primarily a safety study to make sure patients tolerate the drug before moving on to a longer part 2. So part 1 is completed, and the study design is shown here. I'm going to try to make this pointer work. Okay, it doesn't work. Yes, but that's okay. So the study design allows for testing of the isolates from sputum, so up to 6 weeks of time from time of screening to randomization to allow testing of the isolate to make sure there's sufficient Pseudomonas for us to enroll the patient, and also that the isolates are susceptible to our phage cocktail. If that is confirmed and the patient does not have an acute exacerbation during that time period that would prevent them from enrolling, then we randomize them to active or placebo. And in part 1, we had 2-to-1 randomization in a single ascending and multiple dose portion. So the 3-to-1 randomization would randomize to phage or placebo for 7 days on top of standard of care. And we have a single ascending multiple dose type of design. So in the first day, all patients, both in active and placebo arm, received the placebo to make sure they tolerate the excipients. If they did, which they all did, they proceeded on to day 2, and the phage arm received a low dose. And then as they tolerated that, on day 3, the phage arm received a high dose, and then on day 4, they started on twice daily high dose therapy in the active arm, which continued from day 4 through day 7. As you can see by the first line here, the patients had clinic visits on day 1, day 2, day 3, and day 4, and they were on their inhaled antibiotics on the same duration as the phage therapy from day 1 through day 8. Day 8, there was another clinic visit, and there was the last clinic visit on day 15 in part 1, where sputum was collected, FEV1 was measured, and other parameters were assessed. There was a phone call at day 35, and then monthly chart reviews to check for any acute pulmonary exacerbations that happened in the interim, and also an end-of-study phone call at 6 months after the last dose. This is the top-line results for part 1, with actually 9 subjects that were enrolled. We had 7 subjects enrolled on phage and 2 on placebo. The mean age was roughly between 30 to 39, and 5 patients in the active arm were male and 1 in the placebo arm. The majority of patients in part 1 were from Israel, based on the timing of enrollment, and all the patients were white. The mean BMI is shown here, it's about 23, and 33% of the patients were on CFTR modulators, and the breakdown is shown here. The majority of the patients were on tobramycin of the 3 inhaled antibiotics, although aztreonam and colistin were very close, second and third. And the mean percent predicted FEV1 was 66%. And these patients had Pseudomonas aeruginosa on day 1, as shown here, so relatively, I would say, moderate to high levels. Safety wise, the study drug was well tolerated, and there were no premature discontinuations from study drug or from the study. There were no adverse events related to study drug or adverse events of special interest. And we had two types of adverse events of special interest: acute pulmonary exacerbations or a very specific kind of adverse event that was after study drug administration. There was pulmonary worsening after study drug administration, and none of the patients exhibited those adverse events. There were no subjects with clinically significant abnormalities in labs, vitals, or physical exam. And based on the safety data from Part 1, the DMC allowed us to continue to move to Part 2, unanimously approved the study continue. Here is the microbiologic efficacy. So we see here that the mean Pseudomonas CFU reduction at day 15 versus baseline was minus 1.42 log versus minus 0.28 log in placebo. And all the subjects had high levels of screening Pseudomonas at the start, with all the morphotypes susceptible to the phage cocktail, which is an important item to really keep in mind, that these patients were confirmed to have in vitro activity before they received study drug. And bacterial genomic analysis showed that each patient consistently was colonized with the same genotypic strain of Pseudomonas from screening up to the end of therapy. We checked PK in the sputum, so we saw that at baseline, before study drug administration, no patients had phage, which is as expected, and also there was no phage in the placebo arm, which is very reassuring, that these patients either were not given the wrong drug or also did not have endogenous phages that matched the types of phages that we were administering. On day 8, which is one time point we assessed, we had 6 of the 7 subjects on phage who actually had phage present in their sputum, and actually 2 subjects still had phage at day 15. And these are just some of the time points, but basically we had phage detected in the sputum of all the subjects who were treated with phage at any time point, and including some patients 7 days after end of therapy. And there were no phages in the placebo-treated patients and no emerging resistance to the phage cocktail during or after treatment with VX-04A. Okay, now we move on to Part 2. Once Part 1, which was primarily a safety-focused study, was complete, although we assessed efficacy from a microbiologic standpoint, we moved on to Part 2. And here we have a larger sample size. It's a randomization of 2-to-1 with a minimum of 24 subjects on active and 16 on placebo. 24 total with a minimum of 16 on active and 8 on placebo. And the duration of therapy, unlike Part 1, is 10 days of twice-daily therapy. So that is a high dose, as was given in the day 4 through day 7 of Part 1. And we also have later in clinic follow-up. And the majority of the patients, we actually went not just at day 28, but we amended the protocol to allow a day 38, day 38, in clinic follow-up. So that way we could assess, again, a sputum Pseudomonas level and an FEV1. And so far, we have completed dosing in all Part 2 patients. And thank you to all our sites who worked very hard with our CRO to find the patients and get them into our study. So the study design is as shown here. We had, again, a screening period during which we collected sputum and we tested the Pseudomonas for susceptibility to our phage and sufficient levels of Pseudomonas. And then patients were randomized to active or placebo. Again, they had to be on one of the 3 inhaled antibiotics, either tobramycin, aztreonam, or colistin. And the patients were seen at day 1, at day 4, at day 10, which was the end of therapy, day 17, day 28, and then in actually over 50% of the patients, we had a clinic visit at day 38. And then again, monthly follow-ups by chart review to see if there were acute pulmonary exacerbations, and a monthly safety follow-up at 6 months after last dose of study drug. So these are the demographics and baseline characteristics. Most current data is shown here, and this is on all the patients that were enrolled in Part 2. And again, it's interim data because we have not unblinded the Part 2 portion of the study, but the aggregate data is shown here. So we have one column because we have not unblinded yet. So this is the aggregate data, and mean age of 36, roughly half male and female. We had more representation from EU, from our sites in Spain and Czech Republic and Netherlands. And we had also more patients from the US and also a good proportion from Israel. Majority of the patients were white, and the mean BMI is 23. And what's interesting is the percentage on CFTR modulators, of course, indicating the standard of care throughout the world. But it's interesting that 76% of the patients were on CFTR modulators and continue to have high levels of Pseudomonas despite that. So the majority were on ETI, and the other breakdown is shown here. And the majority of patients in Part 2 are on colistin, and then tobramycin is the second runner-up, and aztreonam the third. And the percent predicted FEV1 is 62%, and the mean Pseudomonas burden at screening is 6.5 logs. So in conclusion, Part 1 top line showed that the safety perspective had a very well-tolerated profile for the study drug, and microbiologic efficacy-wise, we had minus 1.42 log of reduction in the active arm versus minus 0.28 log in the placebo arm, on top of standard of care inhaled antibiotics. No emerging resistance in the phage-treated subjects, and PK-wise, there was phage in the sputum of all the BX04 subjects, including up to the day 15 time point. Part 2 is ongoing, and we have similar baseline characteristics with the exception of more subjects from US and EU than in Part 1. Here are my acknowledgements for CFF and IAA. A special thank you to CFF, TDN, ECFS, CTN, and our subjects and investigators and site teams and CRO and vendors and specialty laboratories who worked extremely hard on this study. It's not an easy study to conduct, as I think the sites that have enrolled patients can attest to, and it's I think worthwhile because of what Dr. Solomon mentioned. If we can get at least one patient to breathe easier, it's definitely worth it. Thank you very much for your attention.
If I could jump in, thank you so much. It's so good to see data on this. So I was at a wonderful talk yesterday, Dr. Van Tine, who provides phage on compassionate use. So I'm pretty steeped now in phage hunting and libraries and what goes into creating a phage. So with the commercially prepared cocktail, I mean, what was your screen failure rate? Like, what was the match or likelihood of finding a patient who was eligible for your phage therapy?
So that's a great question. So we haven't published that data. That data is not publicly available, but it will be once we publish the results. Yeah.
I think that was Felix's question. Are there any other questions from the floor? I have one. Any thoughts on what your outcome measure of efficacy is going to be? A microbiological outcome measure is always a little bit dicey.
Yeah, oh, great questions. So we actually assessed FEV1 and CFQR and CFRSD-CRIS and number of acute pulmonary exacerbations all the way through the 6-month follow-up. So that's all data we are now analyzing and will be unblinding in the next few weeks. So more to come on that. But I think that's an important aspect because let's say we have a microbiologic reduction in a patient pool that is 76% on CFTR modulators. Is there room to even get them clinically better, or have they already had the clinical improvement? These are all really important questions, and I think we'll be very keen on looking at the results.
Okay. Dennis?
Thank you for your presentation. A quick question about, the antibiotics, nebulized antibiotics, were they patients on continuous, month on, month off, and were they given exactly at the same time for some of them, or was it on the time of the antibiotics?
Yeah, so great questions. So these were really important parameters we had to wrestle with because we wanted to have a feasible study, but we also didn't want to have too many confounders built in that could assess or impact our ability to assess the results. So patients could have been on any type of cycling combination of antibiotic as long as they were on one of the three from day 1 through day 7 of part 1 and day 1 through actually day 28 in part 2. But they could have been off the month before and then on, and it could have been any combination. However, we wanted to make sure there was no in vitro confounding, potentially if they had just inhaled their antibiotic and then produced a sputum, and then we look at the results and perhaps there could be an in vitro effect of the inhaled antibiotic they just inhaled on the sputum bacteria that's in their specimen. So we wanted to eliminate the confounder. So we told the patients to not take the inhaled antibiotic the morning before the sputum, or at least space it out. That way we minimize the chance of that being a confounder. So that has been incorporated in the protocol, and we urged and reminded sites to do that. And I think that's the best we can do.
Thank you.
Sure.
Thank you for your talk. Did you see a change over time in the sensibility of the bacteria to the phage?
Yeah, so we did not see emerging resistance in the part 1, and part 2 is still blinded, so we're evaluating that.
Thank you.
Sure.
Hi, thank you so much for your talk. Are you planning for a part 3 where you continue the phage for 28 days as well?
Great question. So that would be the next study potentially. So that's what we are currently discussing on how we would approach the study design of the next clinical trial. So the current trial has completed enrollment. It had part 1 and part 2 and it was a phase 1b/2a, but the next trial would potentially evaluate that. That's still under discussion, however. Yeah, great question.
I saw there was some variability in the decrease in the amount of Pseudomonas. And is there any correlation with those that had less would have more severe lung disease, given the possibility that with the nebulized medication distribution, through sort of poorly ventilated areas, excessive mucus plugging, that those may not respond?
That's a great question. We didn't look at that specifically, but I'll bring it back to my team and we'll take a look at that.
And lastly, was there a biosafety concerns at all, or was that a barrier either in conduct at sites or for someone to take this at home?
There's actually no requirement to have phage administered in a certain way or have the phage prepared in a hood, as with some other medications perhaps. And patients actually could take this at home, and they actually did take it at home. They just prepared the phage, basically added contents of each of the vials into their nebulizer and inhaled it without any special precautions. Phage is ubiquitous in the environment, so we don't expect there to be some sort of ancillary effect in the environment.
Okay, well, that concludes our session, and thank you so much, all our speakers.